Good afternoon, everyone. I'd like to just thank you, Carolyn, for the introduction, and thank you to the Physiological Society for giving me the opportunity to present some data today. The data I'm going to present is in two halves, really. I'm just going to give an overview on some of the regulation and limitation of fat oxidation during exercise. It's a big topic, so I'm just going to focus on what we see during high-intensity exercise, because we know exercise intensity is one of the strongest determinants of the rate of fat oxidation. So I'm going to focus on the decline we see during high-intensity exercise and what may regulate that. And then I will also talk about some data from our lab where we think free carnitine availability is limiting to the rate of fat oxidation. So some data where we've manipulated free carnitine availability to provide a bit more insight. Okay, so just three studies here just to show this decline in the rate of fat oxidation during high-intensity exercise. And I'll spend a bit of time on this because this allows us to think about what we mean by fat oxidation when we're measuring fat oxidation. So if I just focus down here on this study from Johan Helge's group who presented earlier this morning, they've done a graded exercise test here in trained and untrained females and looked at the rate of fat oxidation over various exercise intensities. And as you can see in both trained and untrained, during a high, as we increase the intensity from low to high, we get this decline in the rate of fat oxidation. So it doesn't stay at a fixed intensity increase, it's actually actively being inhibited. It wasn't significant in this study, but it looks like trained individuals tend to have a higher rate of fat oxidation and also perhaps have this decline at a slightly higher exercise intensity. But this is measured using indirect calorimetry at the whole body level. And actually, the contribution of the active muscle mass to this fat oxidation is only around 40%. And that changes over these range of intensities. So if we want to know what's happening within the muscle, we need to go perhaps a step further. This study from Luc van Loon, it was supposed to fade in and fade out, is a very similar test where they've had, these are very highly trained cyclists, I think a VO2 max of over 5 litres a minute, exercising at a range of intensities, and where they've tried to quantify substrate sources from muscle glycogen in muscle biopsy samples, plasma glucose and plasma fatty acids measured using a stable isotope approach, so we can actually quantify the rate of plasma fatty acid oxidation. And also get an idea of fat from other sources, which is thought to be predominantly intramuscular fat, but also fat from VLDL triglyceride. And as you can see, using this approach, if we look at the contribution from plasma fatty acid, again, this declines during high-intensity exercise. And also the decline from fat from other sources, so that the total fat oxidation is again inhibited. In order to calculate this fat from other sources, again, indirect calorimetry has been used to calculate total energy expenditure and total fat oxidation, and we quantify plasma fatty acid oxidation and subtract that from total fatty acid oxidation to give us other fat. But nevertheless, it's still using indirect calorimetry. Also, the rate of plasma fatty acid disappearance here using the stable isotope technique, again, only around half of that is actually going into the active muscle. So we still don't really know what's going on within the active muscle in terms of fat oxidation. This approach is the classic one-leg cycling approach that was developed in Copenhagen with Bent Saltine. This is a study with Bente Keynes and Neil Ruderman's group, where they've again looked at graded exercise across the single leg and used an AV balance technique. So that way you can actually determine the fat oxidation going on within the active muscle mass. And again, you see an increase with the onset of exercise in terms of the rate of fat oxidation, but then a decline as we go from a high-intensity exercise. One thing to notice here is the decline comes a lot later. So whereas it's here, it's around 65% VO2 max, and somewhere between 55 and 75% VO2 max. Here it's around 85% VO2 max, probably because blood flow, oxygen delivery is probably not limiting in this single leg model. So I'll go on to why we think this decline is happening, but within the muscle. But first, let's just have a think about potential sites where this could be being regulated. So this is a schematic diagram of a muscle cell. Hopefully you're all familiar with a mitochondria, the sarcoplasm and circulation, and sources of either aerobic in the mitochondria or anaerobic ATP resynthesis from various fuels, fat and carbohydrate predominantly. So what we know during high intensity exercise, there's a slight decline in circulating free fatty acid availability, possibly due to reduced adipose tissue blood flow. And it's thought that this decline in circulating fatty acid availability may contribute to the decline in fat oxidation during high intensity exercise. A study from Bob Wolf's group some time ago now looked at investigating this. So they performed a study at various exercise intensities, so moderate intensity exercise, 85% VO2 max, and you can see the reduction in fat, total fat oxidation at 85% VO2 max, where they also saw a decline in free fatty acid availability. They then replaced it by an intravenous lipid and heparin infusion, and this increased the fatty acid rate of appearance by fourfold, a huge increase in circulating fatty acids, and only partially restored this decline in fat oxidation. So perhaps not the predominant reason why we see a decline in fat oxidation during high intensity exercise. Other potential sites that people think may be limiting is fatty acid transport across the sarcolemma. There's various fatty acid transport mechanisms and transporters. CD36 is one of the more prominent ones. But during high intensity exercise, it's been shown again by Bente Keynes' group that you have an accumulation of acyl-CoA within the muscle cell, and other groups have shown you have an accumulation of intramuscular triglyceride actually during high intensity exercise. So if this is accumulating, it suggests that the limiting factor is perhaps a bit downstream. And so I'm going to present data. The predominant theory is that it happens at the level of carnitine palmitoyl transferase 1, which is the rate limiting step in mitochondrial fatty acid uptake. And the main theory, if you had looked, if I just go back, at the previous slides where we saw the decline in fat oxidation, is you see this acceleration of carbohydrate oxidation. And so the theory is that somehow this acceleration of glycolytic flux is inhibiting CPT1. So just shown here again, this is the same study by Luc van Loon, but I've just pulled the plasma and other fat sources into total fat oxidation. The same study from Bente Keynes and Rudiment, and you see during the high intensity exercise this acceleration of glycolytic flux and a down regulation of the rate of fat oxidation. So a study from Ed Coyle where they looked to investigate this relationship between glycolytic flux and CPT1. They got individuals to exercise a moderate exercise intensity under fasted conditions or when they fed glucose to accelerate glycolytic flux. And then they measured the rate of fat oxidation using stable isotope tracer approach. They used a medium chain fatty acid, octanoate, that is oxidized, thought to be oxidized independently of CPT1, so it can enter the mitochondria independently of CPT1, or a long chain fatty acid, palmitate, which has to be actively transported into the mitochondria via CPT1. And we see that when we increase glycolytic flux with glucose feeding, there's no reduction in medium-chain fatty acid oxidation, but an inhibition of long-chain fatty acid oxidation, suggesting that perhaps this limitation to fatty acid oxidation during exercise is at the level of CPT1. So I'll just talk a bit more about, or focus in now, this is that schematic diagram, but focused in now on fuel sources from carbohydrate and fat, just to talk a bit more about CPT1 and why it's involved in translocating long-chain fatty acids into the mitochondria. So once they're entered into the cell and converted to acyl-CoA, CPT1 esterifies the long-chain acyl groups with carnitine to form acyl-carnitine, and then acyl-carnitine can then be translocated across the otherwise impermeable inner mitochondrial membrane by carnitine-acyl-carnitine transferase, or translocase, into the mitochondrial matrix where it's transesterified by carnitine palmitoyl transferase 2 back to acyl-CoA and free carnitine, and then as we know, the acyl-CoA can then undergo its oxidative fate via beta oxidation. And from the earliest studies of CPT1, it was known that malonyl-CoA is a potent inhibitor of CPT1. And sorry, I'll just come back to, before I say that, just a bit more about the importance of this actual process. If we see in individuals with inherited disorders of carnitine deficiency, CPT2 or CAT deficiency, in those individuals you'll see up to a 75% reduction in the rate of fat oxidation, either in vitro or in vivo. You have lipid storage myopathy, muscle pain, particularly on exercise, hyperglycemia, so an increased reliance on glucose, and hypoketosis. And also, what also highlights this important role of, I think, CPT1 in this process is there's no known deficiencies of CPT1, particularly the muscle isoform, so perhaps suggesting it's incompatible with life. So, as I mentioned, malonyl-CoA appears to be, or is, a potent inhibitor of CPT1. So when we increase glycolytic flux under insulin-stimulated conditions, and you have acetyl co-accumulation, this, via the action of acetyl-CoA carboxylase, goes to form malonyl-CoA that inhibits CPT1 and fat oxidation. So we know this happens at rest, and people thought that it may happen during exercise as well. But what we also know about during exercise is that AMPK is activated, and this is an inhibitor of acetyl-CoA carboxylase. So perhaps suggesting that during exercise this process may not occur. And that study that I mentioned from Neil Ruderman and Bente Keynes actually looks at acetyl-CoA carboxylase activity at a range of exercise intensities, and indeed showed that during the high intensity exercise where we saw a decline in the rate of fat oxidation, there's a decline in acetyl-CoA carboxylase activity, and this was met with a reduction in malonyl-CoA content within the muscle. So showing that malonyl-CoA is perhaps not important during exercise in inhibiting CPT1. So the study from Luc van Loon that was performed in collaboration with Paul Greenup at the University of Nottingham took it a step further and looked at free carnitine availability. So we know that because carnitine is the other, or the main substrate for carnitine palmitoyltransferase 1, and we know that during high intensity exercise we have a reduction in free carnitine availability that matches this reduction in fat oxidation. And it's generally due to this acetylation of the free carnitine pool and an increase in acetylcarnitine. So often an under-recognized role of carnitine is as an acetyl group buffer. It's becoming, there's more and more interest developing at the moment in some disease states, but I won't go on to talk about that today. So this is the role of carnitine as an acetyl group buffer where we have an acceleration of glycolytic flux such as would be observed during high intensity exercise. And the production of acetyl-CoA from pyruvate oxidation via the pyruvate dehydrogenase complex becomes in excess of the demands of the TCA cycle. And when this happens and we have an accumulation of acetyl-CoA, it depletes the very small free coenzyme A pool that's critical for the continuation of PDC flux and also TCA cycle flux. So when this happens, carnitine, via the action of carnitine acetyltransferase, buffers these excess acetyl groups to form acetylcarnitine and maintain this viable pool of free CoA. So if we put this in how important this process is and calculate this flux, you can see these were some calculations that Tim Constantine performed in one of his studies in the 90s. If we assume that maximal PDC flux is approximately 20 to 25 micromoles per second per kilogram of wet muscle during high intensity exercise, the muscle-free CoA pool is only around 10 micromoles per kilogram. Then if we didn't have this process, then hypothetically this entire CoA pool would become acetylated within a second, PDC would be inhibited, and therefore oxidative ATP production would be inhibited. Shown another way, during the first 3 minutes of intense exercise, at a 75% VO2 max, approximately 4,000 micromoles per kilogram wet muscle of acetyl groups are transferred from CoA to carnitine. So if we think of this in terms of the roles of carnitine within skeletal muscle, then Then during high intensity exercise, what we have is carnitine, because the total carnitine concentration is fixed. We have carnitine playing more of a role in acetyl group buffering, and we have an accumulation of acetylcarnitine, a reduction in free carnitine, and therefore potentially a reduction in the availability of carnitine to CPT1. So work that we've been doing in Nottingham over the last 15 years has been looking around manipulating carnitine availability within muscle, depleting or increasing its availability to inform more upon this integration of the integration of the regulation of fuel metabolism within skeletal muscle. So I'm now going to focus more on some of this research that we've done in terms of manipulating carnitine availability, mainly increasing carnitine availability in human skeletal muscle. So some of you will probably remember back in the 80s and 90s, there were a lot of studies performed where people were trying to supplement L-carnitine to have a performance-enhancing effect. Because of this role in fat oxidation, it was assumed that you could increase fat oxidation during exercise and spare muscle glycogen, and it'd have an ergogenic effect or increase exercise performance. But to briefly summarize those studies, there were, this is a summary of around 20 studies. They supplemented anywhere from 2 to 6 grams of L-carnitine for anywhere from 1 day or 1 week up to 3 months prior to a bout of exercise at a range of intensities. And the overwhelming result was that there were no effects on markers of sort of exercise performance or exertion, VO2 max, and also no effect on markers of whole body, fat or carbohydrate metabolism, or even at the muscle level either. But what was clear from these studies was that in the majority, they didn't actually measure muscle carnitine content. And in those studies where muscle carnitine content was measured, it hadn't actually been increased. So clearly, if you want carnitine to affect fuel metabolism, or you want to investigate the role of carnitine within fuel metabolism, you need to alter its content within muscle. The predominant reason for this is because the transport process from plasma to muscle for carnitine, there's a huge concentration gradient, around about 100-fold concentration gradient, and that process is saturated at physiological plasma carnitine concentrations. So simply elevating plasma carnitine availability is probably not going to have an impact on the muscle carnitine store. And we know that if we feed carnitine for 3 months or even intravenously infuse it for up to 5 hours, it doesn't increase muscle carnitine content, and it doesn't increase the net uptake of carnitine across the leg, or I'll show you some data where we've looked across the arm as well. But because we know that this transport process is a sodium-dependent process, and we know that insulin also stimulates the sodium-dependent transport of other similar compounds to carnitine, some amino acids, creatine, for example. Our early work hypothesized that if we could, that insulin may stimulate carnitine transport into muscle. So if I'll just summarize several studies in just this one slide, where we demonstrated that insulin could stimulate carnitine uptake by intravenously infusing carnitine for 5 hours during an insulin infusion. And we infused insulin to maintain a fasting serum insulin concentration, and as you can see here, intravenously infusing carnitine for 5 hours under these conditions had no effect on muscle total carnitine content. This is a supraphysiological plasma carnitine concentration, but still no effect, whereas if we do it during an insulin infusion, we saw around about a 13 to 15% increase in muscle total carnitine content. I won't go into it, but when we increased muscle total carnitine content under resting conditions, it had a fairly profound effect on just resting muscle metabolism. But I'm going to focus on the exercise effects of increasing carnitine. Obviously, intravenously infusing carnitine with insulin is not a particularly practical approach to increase muscle carnitine content. If some of you read the news last year, you'll see that there were some athletes, particularly in the States, that did think intravenously infusing carnitine was a good idea, if you believe the press. So we decided to stimulate insulin release via ingesting carbohydrate. So this was a recent study where we've looked using an AV balance model across the forearm to look at net carnitine balance in response to carnitine ingestion with or without carbohydrate. And just to quickly summarize, so they ingested carnitine here, an hour later ingested either carbohydrate in the red line or just water in the green. And as you can see, around about half an hour later, which had corresponded with the peak in insulin, we saw a net carnitine balance across the forearm. We had done this previously. This is Ben Wall, who's now at the University of Exeter, who supplemented carnitine every day for 24 weeks with fairly large amounts of carbohydrate. So it was 2 times 1.36 grams per day of carnitine with 80 grams of carbohydrate or placebo. Carnitine or placebo, both groups had carbohydrate. And as you can see, after 24 weeks of supplementation, there was around a 20% increase in muscle total carnitine content. So what about the effects of metabolism during exercise? So in that study from Ben, volunteers performed 30 minutes of exercise at 50% VO2 max, then 30 minutes of exercise at 80% VO2 max, and then subsequently a time trial performance test. So this is the data from 50% VO2 max, and consistent with carnitine's role in the translocation of long-chain fatty acids into the mitochondria. We saw an inhibition of PDC activation status, a trend for reduced lactate accumulation, and quite remarkable 50% lower glycogen utilization during exercise. So suggesting that this carnitine-mediated increase in fat oxidation has inhibited glycolytic flux and spared glycogen. In that study, we followed up and looked at fat oxidation. And this is just after 12 weeks at 50% VO2 max, there was a trend for a greater fat oxidation, but what was more clear was actually a significant increase in energy expenditure during exercise at 50% VO2 max. But if you remember, I said during fat oxidation measured at the whole body level, only around 40% of that fat is actually oxidized in the active muscles. So actually gaining insight from carbohydrate metabolism within muscle is probably a more accurate index of what's happening with fuel metabolism. Nevertheless, we went on to use a stable isotope approach to see if we could quantify fat oxidation a bit more accurately. This was actually in older individuals. This is unpublished data. And as you can see, again, 24 weeks of carnitine supplementation. This time it wasn't with a large amount of carbohydrate because it was in older individuals. We gave it in a shake with a smaller dose of carbohydrate and some protein to get that insulin stimulated carnitine transport. Again, in the carnitine group, around about a 20% increase in muscle total carnitine content. This is using the same stable isotope approach that I described in the Van Loon paper, where we've measured fat oxidation from the plasma fatty acid oxidation using a palmitate tracer, and fat from others, and then that gives us fat from other sources if we take away from total fat oxidation. And as we can see, a significant increase in fat oxidation after 24 weeks of carnitine supplementation. This time the energy expenditure wasn't quite significant. And from this, it looks like it's due to fat from other sources, although that wasn't significant. So then we wanted to look a bit further to determine, as I said, this fat from other sources could be intramuscular, could be from circulating VLDL triglyceride, or could just be in other tissues in the body, not actively within the muscle. So we can look at muscle fat oxidation, muscle fat utilization. This is an electron micrograph of a muscle cell showing the mitochondrial network in the intermyofibrillar region and lipid droplets. This is very similar to the work that Clara Prats was presenting yesterday. She was using a fluorescence microscopy approach. But we've used this approach because we can get some idea of the subcellular localization of lipid and what is happening into those different pools. So this is the intermyofibrillar lipid that's next to those contracting myofibrils, but there's also a lipid pool in the subsarcolemmal region. And we're not really clear on if there's any difference in the function of these two pools. So in a previous study, we quantified the utilization of these different pools during exercise. We looked in young, lean individuals, old, lean individuals who were matched for insulin sensitivity, body composition, and also old, overweight individuals. And we can see that, and this is before and after an hour of exercise at 50% VO2 max. In the subsarcolemmal region, there's no utilisation of intramuscular lipids. If anything, it actually accumulates slightly in the older individuals. Whereas in the intermyofibular region, in the younger individuals, we have a reduction in intracellular lipids, suggesting it's used during exercise. I think this raises some interesting questions over the athlete's paradox that was also raised yesterday, but I won't go into that now. And this is where we've actually quantified, using the stable isotope approach, fat from other sources. We can see in the younger lean individuals, they had a significantly higher utilisation of fat from other sources compared to the older individuals. So it corresponds okay, although we didn't actually quantify the intracellular lipid use. So with the increase in carnitine, do we have an increased IMCL utilisation? I'll just quickly go through this, but again, looking at the muscle level in those older individuals, again, at 24 weeks, no accumulation of lactate, suggesting that we do have that upregulation of fat oxidation within the muscle. And it's not clear from this, but perhaps a trend there towards an increased intramicellular lipid droplet utilisation, although this is in older individuals. Briefly, what happens at 80% VO2 max? That was all at 50%, but if you remember, the decline in fat oxidation occurs during high-intensity exercise. So in line with this and the role of carnitine within carbohydrate metabolism as an acetyl group buffer, we saw when we increased, this is going back to the study of Wall et al., increased carnitine, we had around about an 18% increase in carnitine acetylation. And this allowed a better matching of glycolytic to oxidative flux, or glycolytic to PDC flux. This was evidenced by, at the same muscle glycogen utilization, there's a lower lactate accumulation and a greater increase in PDC activation. So a much better matching of glycolytic to oxidative ATP production. And then finally, we asked if there was an ergogenic effect. So after that half an hour bout at 50%, then 80%, they then did a half an hour time trial where they were asked to perform as much work as possible. This was the control where the mean effect, there was no difference. And in the carnitine group, we saw an 11% increase in exercise performance. And importantly, every individual increased. So it looks like having an ergogenic effect. We don't know why, whether this was due to the glycogen sparing effect at low intensity exercise or due to this better matching of glycolytic to oxidative ATP production. I suspect it's the latter because we've subsequently shown changes in cycling efficiency during very high intensity exercise. So just to summarize, the decline in the rate of fat oxidation during high intensity exercise appears predominantly due to a glycolytic flux mediated inhibition of CPT1. We know that skeletal muscle free carnitine availability appears to be rate limiting to CPT1 in vivo, and carnitine availability is reduced during high intensity exercise. But my main message really is that increasing carnitine availability during low intensity exercise has this role in fat oxidation and spares muscle glycogen. But actually during high intensity exercise, it plays more of a role as an acetyl group buffer and allowing high rates of glycolytic flux. And if you consider athletic performance, most elite athletes will be at that level of 75, 80% and above in most of their events. Then having or being able to produce a high rate of glycolytic flux is probably a prerequisite. So taken together, it could provide a strategy for athletic performance. Thank you very much. Thank you for listening.
Thank you very much.